I'm Mohammed Bentires Alj. People call me Momo. I'm group leader at Friedrich Miescher Institute in Basel. My lab focuses on the cellular and molecular mechanisms controlling normal and neoplastic breast stem cells, resistance to targeted therapy, and progression to metastasis. I'm pleased to welcome you to conversations with key figures in mammary gland biology and breast cancer.
So I'm Lothar Hennighausen, and I'm currently at the National Institutes of Health in Bethesda. And I started my scientific career back in 1978 in Cologne as a PhD student. And I would say over the last 35 years, the red thread in my career has been to study the mammary gland, to understand the genetics and how the cells develop, and the genetics of milk production. And of course, we have used different techniques over the time.
Lothar, you obtained your bachelor in Marburg in Germany, in Cologne in Germany. What was your PhD project about?
Well, I worked with a fellow called Sippel, and he had studied lysozyme, chicken lysozyme, and he was interested in domain structures. And he asked me to clone alpha-lactalbumin, which is supposed to have a similar domain structure. And those were exciting days, actually, because a new field had opened up, a revolution, molecular biology, and we started to learn to clone genes. So he asked me to clone this gene. And I tried very hard, and I cloned all the milk protein genes, but never this one. But I discovered new genes, among them the whey acidic protein gene. And it's still my favorite gene, and it has served as a paradigm on how prolactin and hormones control genes in the mammary gland.
And later on, the mice that expressed the recombinase Cray under the WAP promoter were developed. I think your lab contributed to this.
Correct. Back in the late 90s, we realized when you knock out genes, very often the mice don't see the light of the day because the genes are important for overall development. So how can you study such a gene in a specific tissue? And you can do this with the Cre-Lox system.
Absolutely.
And Kai Wagner in my lab at this point decided to place Cre under different promoters which target Cre very specifically to mammary epithelial cells.
So I remember you developed MMTV lines,
Correct,
MMTV Cray
correct.
A and D.
So
Yes.
basically mice which express Cre in stem cells versus differentiated cells.
Absolutely.
And those have become very useful for the field.
So after your postdoc, after your PhD, you did a postdoc at Harvard from 1983 to 1985. Who was your mentor?
Probably most people don't remember this man anymore. His name is Philip Lieder.
Yeah, absolutely.
And he's a very interesting man. He's a very clear and linear thinker. And he only believed genetics. He really didn't believe tissue culture cells too much, because he thought the answer is in the genes. And he used genetics very successfully to make major discoveries. And I would say one key feature about him is that he always followed his interests. Early on, he discovered the genetic code together with Nierenberg, then globin genes, then the first, breast cancer, transgenic mouse. So he always moved on to new fields.
Absolutely. So he was here at NIH before going to Harvard, I think.
But I joined him up in Boston after he moved to Harvard.
And this was before the Bill Muller and before?
Yeah, this was in the early 80s and 83 actually. And Bill Muller, I guess you'll also talk to him one day.
Sure, yeah.
He came in 85 or 86.
Okay, so it's after you left.
Yeah.
So your first paper was on mouse whey acidic protein, the WAP, is a novel member of the family of 4-disulfide core protein. Was this your postdoc work? Can you tell us more about this study?
Actually, no, this was my PhD work.
That was before.
And it was discovery of a new gene.
Yeah.
So in those days, we just made entire cDNA libraries. We sequenced 50 base pairs at a time, and then we discovered many new genes. And the WAP gene was one of them.
So this is different from the next generation sequencing machine that you just showed me in your lab.
Yeah, well, in those days I sequenced 50 base pairs per day, now maybe 50 million.
Absolutely.
And probably cheaper.
Of course.
And healthier.
Yeah.
Because we used P32.
Yes, I know. Yeah. Wow. So in 1982, you published a paper with Jeff Rosen. How did you meet Jeff?
Ah, my friend Jeff Rosen. So Jeff is a very interesting fellow. Actually, he was a full professor at the age of 37. It's very impressive. And I met him in London, in Edinburgh, back in 1979 probably. It was a FAPS conference. And he was a keynote speaker. And I enjoyed his talk, but I disagreed with a lot of his findings. And we have been friends and collaborators since then. But I guess Jeff will probably tell you more about this encounter.
Great. And then in 1985, you joined NIH, so the National Institute of Diabetes and Digestive and Kidney Diseases in Bethesda, as PI and group leader. But before talking about your scientific career, can you tell us a bit about your personal history, birthplace, and childhood?
So I grew up in a small town in Germany called Alsfeld. It's very close to Frankfurt. And this was back in the 1950s and 1960s. And I had a very interesting childhood because my parents worked 7 days a week. It was post-war Germany and it was in rebuilding. So I had a lot of freedom.
Yes.
Not like nowadays where the parents, take their kids to soccer games. I had complete freedom. And that's, I guess, was one of the reasons I joined entered science, because it's a field where you also enjoy your freedom.
Absolutely. So then what made you go into science and not art, music, or sport? If I recall well, you are a racing cyclist.
We'll
I
talk
can
about
even
this later.
see your
Okay.
helmet here in the office.
So, arts and music in my family you just absorb passively. Those are professions where you didn't make any money. But my parents really believed in money. Because, as a post-World War II generation, they believed that you needed a proper job. So they wanted all their children to be doctors. So my parents also were doctors, and then I'm the youngest of 4. And they pushed my 3 siblings into medicine. And when I came up to study, they were tired of pushing me. So I had the freedom actually to pursue my scientific career. And they still kept pushing me, and then I escaped to Scotland. I was for a year in Scotland to study biology.
So you started biology then in Scotland?
No, I started in Marburg.
Yeah.
And my parents still kept pushing me to go into medicine.
Yeah.
And I just said goodbye, and went for a year to Scotland. And actually, that's where I was introduced first into molecular biology with people like Ken Murray.
Yes.
You might recall the name.
The name, yes.
From Genentech, yeah.
Yes.
Early days of molecular biology.
Okay. So then you are at NIH since 1985. Can you tell us about your first years as junior faculty or junior group leader?
Well, back in the day, life was different. So in those days, institutes were not mission-oriented. You mentioned National Institute of Diabetes, Digestive and Kidney.
Yes.
So where does the mammary gland come in? Of course not, but I worked on hormones. So people were more relaxed back then. It was not mission-oriented. And life was a little less stressful for young PIs like me. So I just started by myself. I had no help, and then after 6 months a postdoc, and so groups were allowed to evolve a little slower. And the person who hired me was very interesting. His name is Bill Jacoby, and talking about mentors.
Yes.
Nowadays everybody has a mentor.
Yes.
So he was traditionally my mentor, so to speak. But his mentoring was very different from what you consider nowadays.
Yes.
He basically always said, once a year in our discussion, when we discussed progress, he said, Lothar, publish good papers. And do not join editorial boards. It's just a waste of time.
Distracting you from your research.
Yeah. So those were very clear thinkers.
So who were your role models, if any, and any additional mentors?
So as a child, when I grew up as a teenager, Julius Caesar was my
Julius.
role model. Because he was single-mindedly driven.
Like scientist.
I used engineering
Yeah.
to win wars.
Yes.
Okay, use strategies in engineering, and in science you have to do the
It's
same
about
thing.
this,
yeah?
yeah.
And of course, when I grew up, I had other mentors. But I would say probably the most influential person in my scientific career was probably Phil Lothar. He was such a clear thinker, and he always told me, Lothar, ask key and relevant questions, and then design decisive experiments which give you unequivocal answers.
Yeah, whether it's yes or no.
Yes, and that's where the genetics came
Absolutely.
in. If you mutate a gene in a mouse and there's no defect, then you know the gene is not important.
Yeah.
Sure, there might be backup
Sure.
genes, but this gene is not important. If you see something, then you know
You're
it's
on the
relevant.
right track.
Yeah.
Yeah. So what did your mentor, what did they do that was really appealing to you? Is it their achievement or their communication skills or their mentoring skills? What did they really do that was appealing to you?
Well, as I mentioned a few minutes ago, mentoring had a very different meaning
Yes.
back then. But in the case of Phil Lothar, I would say it was the freedom. So when I arrived in his lab back in May 1st, 1983, so 30 years ago, he just said, Lothar, walk around the lab for a week, look what people do, and then you pick a project. I don't think this would happen nowadays.
Sure.
So when people come to my lab now, they have a certain amount of freedom, but not this vast freedom I have.
Yes.
So in Phil's lab, for example, I decided to work on DNA binding proteins. Nobody else in the lab did this, but he said, well, if you want to do this, go ahead. But of course, the flip side is you had to be successful.
Sure.
So you had to walk into his office with a manuscript at the end of the day.
With figures and...
If you didn't do that, you wouldn't have a paper.
Yes. All right.
Again, big changes from today.
Of course, that's a huge
Yeah.
change. So from the beginning, you were interested in milk proteins and their regulation. What did we know on this regulation during the early 80s?
May I come back to the mentors?
Sure, of course.
I forgot to mention my parents, who've been dead for quite some time already, but I think they were also good role models.
Yes.
So they worked all the time
Yes.
and gave me a lot of freedom, so to speak.
Yeah.
I was unsupervised. But they also taught me, by working hard and having 4 children, that it is possible to combine a career with having children.
Yes.
And my wife and I, we have 3 children. We actually followed this path.
So they were mentoring by the example
Yes, yes.
because
yes.
they were working hard and yet they managed to have
Correct.
children.
Correct, yeah. So now you ask, it was
So from the beginning, I mentioned that you worked on the regulation of milk proteins.
Mhm.
So what did we know about the regulation of milk proteins back then? You said you were interested in proteins binding to DNA. But what specifically about milk proteins? What did we know?
So in the late 70s, early 80s,
Yes.
we know very little. This was basically before gene cloning. What we knew at that point was that if you take mammary gland tissue and culture it with various hormones, you can induce those genes. But the mechanism was completely unknown. And I think the advent of transgenic animals, which again was in the early 80s, changed the game, was a game changer. So we could then take promoters of genes
Yes.
and link them to other reporters and evaluate their importance in transgenic mice. And it became clear from our work and Jeff Rosen's work back in the mid to late 80s that all the regulatory elements for milk protein genes are located in promoter regions, in enhancer regions. So the cell-specific elements
Yeah.
and the elements which control the induction during pregnancy by prolactin. That's what we learned in the 80s.
Yes. Yeah.
And then another game changer was, of course, the identification, the cloning of the transcription factor.
Yes. We will come to one
Okay,
key
good.
one. So this was actually STAT5.
Yes.
And Can you tell us more about the discovery of this important transcriptional factor?
So that's actually a very personal story. So Bernd Groner,
Yes.
I hope you have a chance to interview him one day. I met him at a conference in Scotland over a lot of whiskey back in maybe 1994. And he had just identified a new protein called mammary gland factor.
Sure.
Because it was enriched in mammary cells.
Absolutely.
And he had a cDNA. And we discussed this at this meeting, and we decided this was important. And then I had a Chinese postdoc start in my lab, Shuwen Lu, who probably was the most driven person in my lab ever had. And I said, Shuwen, Bernd Groner has the cDNA from sheep.
Yeah.
Let's clone the mouse gene. Let's knock it out and see whether it's important.
Yes.
So MGF then turned out to be STAT5.
Yes.
Signal transducer and activator of transcription, STAT5. And of course, as you know, and maybe later we talk about this, the prime function was in the mammary gland, but now it's been shown to be very important in many other tissues.
Including the hematopoietic system. We will come.
So those were exciting days. We had small labs, but technologies were moving so fast, and we had to jump on the bandwagon, and one could pull it off.
Do you remember why it was cDNA from the sheep? Because that's...
Of course, in the olden days, when you try to clone a gene, you had to use a means to get to the gene. And it was a protein, of course.
Absolutely.
So he had to isolate lots of protein.
Quantity.
And I think he isolated MGF, actually Hiroshi Wakao in his lab, from 5 kilograms.
Yes.
Which is, for the Americans, probably 11 pounds of mammary gland tissue.
Okay. Yeah.
That's my sheep.
Yes, exactly. So
and then
we went back.
And then we made libraries from the mouse.
Yes.
And Shu Wen in the lab, he identified STAT5A and discovered a new isoform, STAT5B.
Yes. So in 1995, you published with Gertrude Robinson and Gil Smith a paper entitled Mammary Epithelial Cells Undergo Secretory Differentiation in Cycling Virgins, but require pregnancy for the establishment of terminal differentiation. What was it about?
I think we should give credit to the people who did the work. Can I get Traudel and she can talk to the camera for 30 seconds?
Absolutely.
Because I just provide the money in life and some ideas.
Absolutely.
Okay.
This is fantastic.
Let me first introduce Traudel Robinson. She's from Austria and she joined my lab in 1993. And Traudel actually probably had a bigger influence on many people than I had. She is the world's expert on mammary gland transplants, especially embryonic transplants. So, for example, why do we do transplants? If a mouse, a knockout mouse, dies in utero, you cannot study mammary gland development.
Absolutely, yeah.
Correct, Momo?
Yes.
Okay. So the way to bypass this is you take the Anlagen of the embryo and transplant it into a cleared fat pad.
Absolutely.
But ask Traudel the question, and she will tell you exactly why she did this.
So in 1995, you published with Lothar and Gil Smith a paper entitled Mammary Epithelial Cells Undergo Secretory Differentiation in Cycling Virgins, but require pregnancy for the establishment of terminal differentiation. What was it about?
Well, we know already for a long time that during pregnancy, milk protein genes get turned on in a certain temporal sequence, and we wanted to look at this on a cellular level, with really looking at the organ in situ. And one of the two things that we found that were very surprising, that actually the milk proteins do already become activated during the cycle, during stimulation by hormones, but if there is no pregnancy following, then this goes away. And the other thing that was really surprising is that we could see that in pregnancy then, when the milk protein genes stay on, that not all the cells in an alveolus were synchronous, and even though they were all bombarded by the same hormonal systemic stimuli, they responded in a not uniform fashion.
Oh, that's fantastic. And later on, using the WAP-Cray system and beta-gal staining, K.Y. Wagner from your lab with Gil Smith identified these cells has been present, of course, in the virgin mice during the cycle. I think it's 1% or 1 to 3%
Yep.
of the cells. Oh, that's fantastic. Thank you very much.
And
Thank
actually,
you.
this paper is a highly cited
Cited,
paper. And
yeah.
yesterday, Traudo just told me, "Wow, I'm surprised this paper can still get cited." Because it's 1995.
Yes,
And in this short-lived world, who reads this old stuff?
absolutely. As we were speaking earlier, what matters is what resists the test of time. And I'm sure these kind of papers will resist the test of time. Thank you very much.
Well, yeah, okay. But the other response with the embryonic transplants, I mean, it's really a very powerful system to take tissues from mutations that are embryonic lethal and be able to transplant them into.
So, who taught you the transplantation assay?
That was Gil Smith.
Okay.
I brought in the knowledge that I brought in was that I knew where exactly all those Anlagen are in the embryo, which is very intimidating to many people.
Yeah.
And I had learned how to dissect them during my PhD thesis. And Lothar's idea was, well, let's try how this works when we transplant it into adults. And
Thank you. So in 1996, you published a science paper entitled Time Sensitive Reversal of Hyperplasia in Transgenic Mice Expressing SV40T Antigen. Polyploidy was reversed at 4 months of age, but not at 7 months. How would you interpret this observation with all what we know today?
Well, let's take a step back. I mean, again, those were very exciting times. And Priscilla first, my wife, and I went on sabbatical to Peter Krus's lab in Göttingen. You might know Peter Krus.
Yeah.
And when we arrived there in 1992, Hermann Bujar just had published the TET system.
Okay, yes.
And we read this paper and said, we have to do this in the mouse. That's the first time we can actually turn genes off and on in the mouse.
Absolutely.
So we got the clone, and Max Planck had all the money. We could do the experiments very quickly.
Yes.
So we generated the first transgenic mice, which allowed us to activate genes deliberately, inactivate them. And this study, which was a collaboration with Priscilla Firth, clearly showed that there are two steps, a reversal steps and an irreversible step. And that was a new aspect. This was the fascinating aspect. And since then, Priscilla Firth has followed up on this and discovered the genetic mutations which are part of the second step, which is irreversible.
That's fabulous.
But it was, again, exciting times because new technologies come by.
Yes, of course.
But you have to jump on the bandwagon and you have to have the money to do it.
To do it, yes,
Yeah.
absolutely.
And the freedom. And again, this was my sabbatical. And Peter Kruse actually also might be a role model in that case. He always gave people freedom. And he said, Lothar and Priscilla, I give you a technician, I give you money. If you believe in this, do it.
Do it, absolutely. So then you contributed many studies on the effect of prolactin and STAT5 on mammary gland development. Can you tell us about your major conclusions?
Well, the major conclusions are that a single hormone can control different aspects of development. So, for example, if you look at the mammary gland, first you need the proliferation of cells, the alveolar cells. And secondly, those cells have to differentiate to make milk. Two very distinct aspects. And a single hormone can do both.
Yes.
And recently, Traudo Robinson and Daisuke Yamachi in my lab discovered that this is done by different concentrations of STAT5.
Absolutely.
So you need very little of a transcription factor to control proliferation, but in order to induce differentiation, you need to boost the levels.
Absolutely.
Okay? So this was the exciting thing. But there's still many questions open. So, for example, we performed genome-wide analysis of STAT5 binding to all the genes, to the entire genome. And we recognize, of course, that milk protein genes bind STAT5 because they're activated by STAT5.
Yes.
But about 90% of the genes which bind STAT5 are not under its control.
Oh.
So are those just parking lots for the transcription factor? So I think the more experiments we do, especially large-scale genomic experiments, the more questions we have.
That's fascinating because there are a lot of questions to be answered by the next generation of scientists.
Mm-hmm.
So the mammary gland also secretes prolactin. So what are the implications of this secretion for cancer? Do cancer cells also overexpress prolactin? I think it's the case.
So it's quite clear in the mouse at least that prolactin can drive proliferation of normal cells and cancer cells. The question only is that is it relevant for human disease?
Yes.
And I think there the jury is still open, because the mouse clearly is different from human.
Yes. In terms of prolactin.
Yes.
So, but in adults, I think there is a level, in some adults, there is a level of prolactin that is found in human.
Correct, right. But I think what we would need to do in that case, and again we can use mouse genetics,
Sure.
we would need to specifically inactivate the prolactin gene in mammary epithelial cells.
Yes.
And then we could study the autonomous effect.
Absolutely, or alternatively to express it specifically in the mammary gland.
Overexpress
and
it,
then
yes, correct.
And
But you're right, specifically in mammary
gland,
epithelial
and
cells.
then
Yeah.
check the effect of prolactin. So after all these years of studies on prolactin, what else needs to be known on this important hormone?
Again, I think from the protein level, we know a lot already how the hormone binds the receptor and passes on its signal to the transcription factors. But what we do not know yet is the cell specificity a hormone has.
Yes.
And the cell specificity most likely is not on the level of the receptor. Like the prolactin receptor can be found on many cells.
Sure.
But milk protein genes, the key targets in the mammary gland, are only expressed in mammary epithelial cells. So for me, the next frontier in this field is the understanding cell specificity. Why are mammary-specific genes expressed only in the mammary gland
Absolutely.
and not in liver?
Absolutely.
In liver genes only in liver, not in the mammary gland. But both are under control of STAT5.
Yes, absolutely.
So I think we are moving probably as a scientific field more into the arena of chromatin, maybe even histone modifications.
Sure. Epigenetic regulation.
Correct.
Absolutely.
So does epigenetic regulation control the ability of transcription factors to come in, bind to genes and activate them? And exactly that's what we're doing right now. We're studying histone modifications and how this affects hormone-induced gene expression.
Well, that's, I look forward to seeing these results.
Well,
But
if they ever get accepted.
Of course.
As we discussed earlier, nowadays it might take 2 years before you get a paper accepted.
Clearly.
Whereas my first paper, as I
Yes.
mentioned to you earlier, was accepted within 2 weeks, back in 1982.
Absolutely. Absolutely. This can be discouraging, of course, for graduate students and for postdocs.
It's devastating. So as a matter of fact, many years ago I had a summer student in the lab. And at that time we had submitted a paper to Nature Genetics. And we went through several rounds of reviews. And when the summer student read the reviews, he said, "You guys are grown people. They treat you like children. They infantilize you.
Absolutely.
I will not go into science," he said.
Wow.
He's a very successful lawyer now.
Okay.
But there's some truth to it. And eventually
Absolutely.
we withdrew the paper from Nature Genetics, submitted to Molendo, and it's been cited now more than 200 times.
Again.
So again, for young people,
Yes.
if you can wait long enough, it doesn't really matter where a paper is published.
Yeah.
If it's solid, it will get cited.
Absolutely. Absolutely. It will resist the
Yeah.
test of time.
Yeah.
Absolutely. So back to prolactin. Prolactin receptor is found overexpressed in human breast cancer, and I think there are clinical trials with inhibitors of the prolactin receptor. What would be your prediction based on?
Well, you should get a paucity in proliferation, but I guess we will see the data pretty soon.
Sure. So Lothar, during your career, you generated several mouse models of mammary cancer. What would you advise a PhD student or postdoc who would like to generate such models today?
I would be hesitant. It takes a long time to generate mouse models. And you mentioned to me earlier, you had a 5-year postdoc and it was a typical mouse genetics project.
Yes.
But times are moving on. If you look at the funding situation, if you only have a mouse model in your application, you will not get the money most likely. I think everything is moving more towards human specimen, primary specimen from cancers, studying them in 3D culture, maybe even in nude mice. So I think times have changed.
But yet, even with the primary derived xenograft, PDX, I think these are important tools, but we still lack the immune system, when we transplant them as xenograft. So I think, don't you think that transgenic models will still be very useful as allograft, even for testing inhibitors, or by identifying the function of these genes?
I fully agree. But on the other hand, if you look at BRCA1, for example,
Yes.
clearly you know that human to mouse differences. So one can use the mouse as a starting point, but eventually you have to back it up with primary human tissue.
Absolutely.
But if you would submit a grant only with mouse, you might have a hard time. You have to bring in the human.
Sure, to confirm what
Yeah.
we observed. Yeah. So, Lothar, more recently you studied the effect of STAT5 in hematopoietic cells.
Yes.
What triggers this shift, and can you tell us more about this effect?
So when you generate a knockout mouse, you have to get, of course, the biggest bang for your buck. So if you only have a mammary gland phenotype, sure, you study only the mammary gland. But if you realize, there are other defects, and we found defects in the liver, in the knockout mice, in the hematopoietic system, of course, you pursue those. Not only because you get additional papers, which is always helpful, but also by comparing the loss of a transcription factor in different tissues, you will learn something about the cell specificity of this transcription factor.
Yes.
And we also learn something about the commonalities.
Yes.
So, for example, what we have learned in those studies is that maybe there are 70 genes which are under STAT5 controlled in every single cell,
Oh, that's
including SOX2 and
Yeah.
CSH. And then on top of this, you have all the cell-specific genes.
Yes.
Milk protein genes in mammary gland, cytochrome P450 in liver, immune globulin genes, and then you can base in transferrin receptor in
Yes.
the lymphocyte lineage. And this allows you then to study the cell specificity.
Okay, yeah, that's absolutely
And it allows you to make collaborations and make more friends.
sure and more
So,
fun. Science
yeah,
is fun.
Yeah. And I think that's actually what the young people have to learn. They have to learn to come out of their nutshell, out of their shell. Okay?
Yes, that's right, yeah.
Instead of just working with an ice bucket in the lab by yourself, if you see a phenotype, let's say in T cells,
Yes.
you pick up the phone and you call John O'Shea at NIH or Warren Leonard. We have so much expertise.
Yes, absolutely.
And actually within days you have collaborations.
Absolutely.
And actually Shuwen Liu I mentioned earlier, he
Yes.
was a paradigm for this. So when we first generated the STAT5A knockout
Yeah.
mice back in 1995, we started, we had in Building 10, Warren Leonard, John O'Shea, those are
Yes.
famous immunologists,
Of course.
and they lined up with ice buckets. So Shu Wen killed a mouse,
Then-
first took out the mammary gland, then the bones, and the spleen, then the liver. So that's how you're efficient.
Absolutely, absolutely.
And that was the early days of team science.
Yes, absolutely. I think this is so important, and I think this is a message for the next generation of scientists. Absolutely.
So basically enhance the value of your research.
Research, absolutely. So you mentored many students and postdocs that are now successful PIs. How important was this for you?
They're the lifeblood of science. Without them, we would not be sitting here and talking about this. And I could tell you a story by each of them. And each of them contributed very significantly to my career. Peter Gassal, who is now director in Scotland,
Yes.
and Kai Wagner, Shuwen Liu,
Yeah.
many.
Yeah. So it reminds me of a saying by the French philosopher Montaigne, who said that teaching is not about filling a vase, it's about igniting fires.
Mm-hmm.
Does this quote? How do you understand this saying in light of your mentoring
Well,
of
in
students?
order to light a fire, you have to have some fuel as well.
Absolutely. So
And of course, some postdocs have more fuel than others. Okay, and some already come being on fire.
Yes.
Okay, and some you try to light, but doesn't work. Okay, but you're right. It's like this. But I think it's always the interaction. So the most successful postdocs in the labs were those who had their own ideas, who came to me and said, I would like to do this.
Then you give them the freedom. The way your
Correct.
mentor gave you the freedom.
Basically, I just advise them. I say, this might be a good idea or a bad idea. But that's a nice thing about NIH, I did not have to write grants, and I only have to justify my research after the fact.
Of course.
So we were actually able to switch directions many times.
Yeah, that's fascinating.
But in the end, it's always driven by the postdoc.
Yes, and creativity and the work to test the hypothesis. Yeah, absolutely.
And the willing to put the extra hours in.
Sure, absolutely.
Because the number of publications correlate with the number of hours people spend in the lab.
Absolutely.
I had this one postdoc, Keiko Miyoshi, from Japan, back in the early 2000s. She studied STAT5 knockout mice, and she was in the lab all the time. She only took time off for the haircutter once in a while. And I asked her, I said, Keiko, you have to sleep sometimes. She said, I can sleep when I'm back in Japan.
So, what would you advise a junior faculty who is going to start her or his team to work on mammary gland development or breast cancer?
That's a very difficult question. So, when we worked on development in cancer back in the 90s, the field was not very crowded. It was very easy to obtain for Marta Gallego in my lab a PhD grant, because not many people studied this. But the field is extremely crowded. And people enter the field now, Momo, they have to compete with you. And who can compete with you? Okay? Who can compete with established people? So I think it's more difficult now to enter this field. And probably I would advise scientists, unless they're really dedicated to the subject, I would say don't enter the mammary gland field.
But mammary gland field is fun. I go to the Gordon Conference on mammary gland now since more than 10 years. And it's one of the best scientific communities I know of. People are very welcoming. I think, you can go to the meeting and sit and realize that next to you, you are sitting to key figure in your field, and then they just introduce themselves, and then you can start fantastic discussions.
Yes, and it's a fantastic field, I fully agree. But when you look at, for example, breast cancer as a disease, I think physicians have a pretty good handle on breast cancer. If it's diagnosed early enough, the survival rate is pretty good.
Yes.
Other cancers, like pancreatic cancer, ovarian cancer, are far more deadly.
Absolutely.
And I think funding might move more in this direction.
And then make it harder than for people who want to enter the mammary gland
Yes.
field.
Yeah, people always say that mammary gland is overfunded.
Yeah. But yet, despite all what we know about breast cancer, there are more than 1,100,000 women that are diagnosed with breast cancer, and from these women, more than 450,000 will die from their disease, despite all the new therapies and our understanding. So there is clearly a need for more better therapies for metastatic breast cancer, specifically triple-negative metastatic breast cancer. Don't you think?
I fully agree. But there's also a realistic budget aspect.
Yes.
So if you would enter as a young PI now and write a grant in the breast cancer field,
versus
you might have a harder time getting this money rather than writing, on other diseases. Yeah.
So, Lothar, what are the major questions that you think still need to be answered in the field of mammary gland development and cancer? Is it metastasis?
Well, I'm not a cancer person, so I leave this up to my wife. The development, we're still struggling, not just we, everybody, with the enigma about cell specificity. And I think in the next 5 years, major progress will be made in this field.
You mean that lineage tracing will help us answer some of these questions?
Possibly, but I think the rapid development of more reliable primary tissue models and transgenic animals where we can test elements and their functionality will really help.
Before we come to the last questions, I would like to come to the cycling aspect.
Cycling.
I know you did Paris-Brest-Paris. Can you tell us more about this hobby? I know you spent some time on this.
Well, back in 2002, I went on sabbatical to Munich. I was a Humboldt professor at that time, and I worked in Axel Ulrich's lab in the Max Planck Institute. and me and my children went to Munich. My wife commuted at that time, and we decided to take a year off. And I discovered cycling as my passion. And as I went on, I cycled longer and longer distances. And now I do those ultra long distances, up to 1,200 kilometers. And if you think about it, so it's called randonneuring, this sport. And we have time limits. So you have to cycle 1,200 kilometers in 90 hours. 90. This includes, sleeping breaks.
Yes.
If you think about it, it's the same qualities as you need in science. You need for this.
Yes, absolutely.
You need strength, of course. In science, you need skills, pragmatic skills, but you need dedication.
Yes.
And perseverance.
Perseverance, absolutely. Yeah. Absolutely. I see that you have the the
See?
This is fantastic.
Those are the signs on the road. But you need perseverance, and that's what I have to tell the young people.
Yes.
Don't give up.
Yes. But then also, after the mountain, there is always a hill.
A downhill.
A downhill.
Yes, yes. So you have to cycle uphill, it takes a lot of energy, but then you can cruise downhill. Like when your paper gets accepted.
Absolutely.
That's the cruising downhill. You can enjoy this and recover.
Yes. So, Lothar, we are coming to the end of this discussion. What would you like to tell the next generation of scientists who will watch this video?
First of all, we always say, follow your passion. Okay,
Yes.
and that's a very, easy thing to say. I think what young people have to do is they have to look at opportunities which open up. Okay, and you have to match somehow those opportunities with your passion. And the problem right now is that many people in my generation, we feel very much empowered. We do not want to retire. So people in the 70s, 80s, and 90s still work. And this, of course, then severely limits the opportunities for young people. Talk to Jeff about this, okay?
I will.
And this, of course, happens with every generation.
Yes.
But I was blessed and fortunate that I grew up in a generation where a whole new technology was established, molecular biology. And old people couldn't do that. So young people were able to drive those people out of business.
Yes.
Because the old people didn't have the skills. And I think that's the same thing now. Young people should look at skill sets, at developing skill sets,
Or niches.
which allows them to compete with established investigators.
Yeah.
Because look at us. We have published a lot of papers. Yours are more high impact than mine, but we have published a lot of papers. And for young people to compete with us, it's very difficult.
Yes.
Because we have the technique set up and
Yeah,
everything.
the lab setup, absolutely.
So they have to do something completely novel.
Yes, absolutely. And that sees the opportunities.
Correct.
Yeah.
And
Much-
I can see actually bioinformatics or information
Absolutely.
technology as one of them.
It's
For us, it's hard to learn all the algorithm.
yeah,
But the young people, they can make an impact.
absolutely.
The other thing is,
Okay.
and when you are a postdoc, just take the fate in your own hand
Yes,
and work on something which you can take with you.
absolutely.
Make it clear to your boss or
Yeah,
mentor right from the beginning, that you're working for yourself.
Yes.
Okay?
Okay. So, Lothar, is there any question that I should have asked, but I didn't?
I cannot recall. Cannot think of any.
Okay, so it was pleasure.
Okay,
talking with
thank you. Yeah.
Thank you very much.
And you probably wonder why I bounce all the time.
Yes. Oh,
Let
yes.
me show the camera.
Let me.
I sit on a ball.
I
Okay. This really is good for my back. So my wife told me several years ago I have to work on my core strength.
So you use the ball all the time?
Yeah,
for,
I said, we have balls at home at the dinner table. We don't have any chairs anymore.
Oh,
Well, we
that-
have some chairs they're stacked
Yes,
away,
for-
but the whole family sits on balls.
Okay. Thank you.
Yeah. Okay. And maybe I can show you this picture. This Philida in the front.
Oh, wow. So when was this picture?
This
taken?
was 1983.
Oh, wow. At Harvard, right?
Yes.
Oh, this is in the quad.
Yes.
Oh, wow.
Oh, you recognize it?
Of course I recognize the quad.
Yeah.
Oh,
So how many
fabulous.
years were you in Boston?
5 years.
5 years.
So yeah, of course.
I cherished my time. Those were exciting times.
Absolutely. Absolutely.
Again, we had a lot of freedom. And I think nowadays people have less freedom. Because when you write an NIH grant nowadays, you have to execute the aims.
Yes.
Back then, when Philida wrote a grant, he was given the money to do whatever he deemed to be important.
Thank you very much.
Thank you, Momo.
Thank you.
So whenever new postdocs come to the lab, I always show them this plate. And it's a German saying. And Momo, you speak German?
A little bit. So
Jeder ist seines Glückes Schmied, which means basically everybody is responsible for their own destiny. That's where I show them to them.
Absolutely.
And they look at me with big eyes. Okay, thank you.
Thank you.